Okay, so I'm Claire, and I'm going to talk to you about how simple sugars might be able to combat bacterial infection. And to do this, I myself, I'm a cell biologist, but we've combined with chemistry folk to help us in this endeavour. So cilia are these beautiful hair-like structures that line our airways, and their motile activity in one direction allows an escalator to move your mucus up and out of your lungs to clear your airways of invading pathogens, bacteria that we inhale, and other things. And I'm interested in what happens when these cilia go wrong. So in the normal circumstance, you would have the motion, but in response to a bacteria here, the bacteria can make proteins called lectins that are capable of binding to sugars on the surface of the cilia, rendering them completely static and allowing the bacteria to settle on the surface and form a network known as a biofilm. So we propose an intervention where we use external sugars that have been chemically modified that will intercept these bacterial proteins before they settle on the cilia, therefore allowing the cilia to retain their motion. And in this way, we might be able to allow patients to defend their own lungs using mucociliary clearance. So a particular infection that we're interested in is Pseudomonas, and here you can see Pseudomonas settling on airway and forming a biofilm, which increases its resistance to antibacterial agents. It's particularly problematic. It's ubiquitous in the environment, so it's in soil and earth, and it's in plant and animal tissues. But it's a particularly nasty, opportunistic human pathogen, affecting patients with respiratory problems, patients that have immune systems that aren't working so well, newborns, patients that have had burns and their skin is impaired and wounded and get colonised by this Pseudomonas. And a systematic review looking at 13 years in European intensive cares found that 17% of cultures from intensive care actually were positive for Pseudomonas. And over 400,000 patients in Europe were found to have an antibiotic-resistant strain. So our idea of sugar therapy isn't particularly a new one. Our researchers at Southampton have already shown that Pseudomonas lectins that stop cilia can actually be treated using fucose, a specific sugar that can intervene in this process. And patients with cystic fibrosis, so they have impaired lungs, have inhaled sugars with no adverse effect and seen a reduction in their Pseudomonas burden. So for our project, we sample the airway epithelium by taking a small brush into the nose and acquiring cilia from the juicy part here on the inside of the nostril called the turbinate. We can then take those cells and we can interrogate them using high-speed video microscopy. So here we've got a high-speed video camera, an environmentally controlled Perspex case, the sample's put here and we get a readout here. And we can record up to 500 frames per second, allowing us to take high-speed videos and then slow them down to look at how the cilia are working. Here you can see in a normal setting, the cilia are beating. And where the cilia are beating, we can use computer software to work out how fast they are beating. And depending on this rate of beating, a colour is assigned to the pixels you can see here. So the areas of black represent no movement. And in response to lectin, this is the normal circumstance, in response to lectin, just 1 hour of treatment was enough to render the cilia less motile or even static. So here you can see in the normal setting, the cilia are capable of beating over a 24-hour period. However, in response to lectin, so this is the normal setting here, but after 1 hour, you see a dramatic reduction in their cilia motility. And this is sustained throughout the rest of the experiment. However, if you pre-incubate the lectin, the bacterial lectin with a sugar, a specific sugar, you allow the cilia to retain their motion throughout the course of the experiment. Therefore, we're showing that this is a protective treatment. So I keep talking about sugar. I don't actually mean any sugar, and this is the table sugar we'd expect to see on our cornflakes or in our coffee. This is actually made up of glucose and fructose, which makes sucrose. And actually the sugar I'm interested in is fucose. And fucose, here, our chemistry partners have been clever enough to be able to modify aspects of our fucose molecule to enable it to be a more potent binder to the bacterial lectin. So future directions for our research. So we've already shown that a potential protective effect of our basic fucose, but we aim to look at how the modified fucose will be, whether it's a more potent protector of ciliary function. And in keeping the cilia motile, we're hoping that this will actually prevent the bacteria from settling on the airway surface. And because they won't be able to settle on the airway surface, we expect that this might improve antibiotic efficiency. So if the bacteria can't make their network, then antibiotics may be more successful at clearing them, and the cilia may be more successful at moving them away. And hopefully these sugars will become prototypes for an aerosolized therapy. But it might not just be aerosolized therapy, it could possibly be used as a rinsing strategy for upper airway infections as well as lower airway infections or other therapies. And so I'd now like to thank the biology team. I'd like to thank expertise from the microbiologists and importantly our chemistry collaborators without whom we wouldn't have been able to do our study. And NAMRIP for our funding. Thank you very much.